Okay. This is going to be flower color. So I want you to tell me, and this is going to be dominant, recessive, right? So using R is red, W is white, R, red is dominant. How many red flowers will there be, and how many white flowers will there be? 3 red? Okay, right? So it would go okay, everybody see that? Just want to make sure because there's going to be these kinds of problems. Okay. We went over this a little bit, but I want to just introduce you to something. There are other kinds of inheritance, ways to inherit stuff. So besides dominance and recessive, as we would see over here, you'd end up with 3 red and 1 white. But you can also have something called incomplete dominance. And so in that case, you can actually, it's more like blending. So you can have 1/4 red, 1/4 white, and 1/2 pink. Okay? And then to make it even more confusing, you can have something called codominance. Where you have flowers that are red and white. Okay? So that's codominance. Incomplete dominance is where it's something in the middle, in between, whether it's color or whatever it is. And then codominance, you have both. We already did this one. Okay. And then to make things even worse for you, so I've been telling you that sickle cell anemia it's dominant and recessive, but it's heterozygote superiority. So remember, we're talking about different environments here and where certain kinds of hemoglobin are favored. Okay, so if you have malaria that's present and you have wild type, normal hemoglobin, HbA, HbA, this is where these individuals get infected with the Plasmodium parasite, as you can see. A lot of these red blood cells have been infected. Eventually they're going to burst open, there's a long life cycle, you don't have to know. But that's what it looks like. Now, when malaria is absent, oh yeah, so in this case, this is low fitness, okay? Because they're all getting slaughtered from the malaria. Okay, malaria absent. So regular red blood cells, they carry the most oxygen. Okay. Now, when you have the heterozygote, and malaria is, well, let's take a look at when it's, when malaria is absent. For the heterozygote, or for the homozygote, obviously we have sickle cell anemia. Also, HbA, HbS in malaria absent would have lower fitness than the regular hemoglobin. Because the regular hemoglobin carries more oxygen than HbA, HbS. Now in the past I've just said HbA is dominant to HbS, but it's more complex than that. I just want to give you guys an idea of what we can actually see out there in terms of inheritance. And then finally, what actually happens with HbA, HbS, the heterozygote, is you have a malaria-resistant phenotype. That means malaria parasites have a hard time invading the cells. However, and that's why it's heterozygote superiority. However, what we actually see here is both types of red blood cells. We have some that show sickling, some that show normal. You don't have to worry about these. These are just immune cells. But they're still kind of resistant to malaria. So in that case, we actually have incomplete dominance because we've got both. It's like red parts to a flower, white parts to a flower. We've also got codominance, or rather, yeah, incomplete dominance, where it's kind of halfway in between. So I won't necessarily have this on the exam, but there's a lot of things that can go on in terms of inheritance patterns. And let's say I had a situation where we had people who had grown up, whose ancestors had grown up in an environment where there's malaria, and there was a fairly high incidence of HbA, HbS, the heterozygote, and HbS, HbS, sickle cell anemia. And then the descendants, you put them in an environment where there's no more malaria. Would you expect the heterozygote HbA, HbS to go up in frequency or down in frequency? Remember, we're in an environment now where there's no malaria. Huh?
Up in frequency?
No, down. So the highest fitness in this, in an area where there's no malaria, is normal red blood cells, HbA, HbA. Unfortunately, HbA, HbS carries, it has some sickling, and it carries slightly less oxygen compared to the dominant phenotype. So in that environment, where there's no more pressure from malaria, you would expect that genotype and phenotype to go down in frequency. And the normal frequency, the normal dominant HbA, HbA to go up. Does everybody understand that? I just want to make sure. I mean, if I were to reverse it, and I said, okay, I take a population that is suddenly exposed to malaria, what genotype and phenotype would you expect to rise? Would it be HbA, HbA, or would it be HbA, HbS? Well, you'd expect the malaria-resistant phenotype to actually increase in frequency. Okay? Okay. We're just starting with sex chromosomes. So humans have, of course, 23 pairs of chromosomes. 22 are autosomes. And then there's a pair of sex chromosomes. And an individual who's diploid either has an XX or XY. So the Y chromosome itself has genes on it that do not occur on the X chromosome. And what it does is it signals the production of certain hormones that then make it male during fetal development. Now, you can also have disjunction, like I talked to you before about trisomy 21, where the individual inherited 3 chromosomes instead of 2. Okay? And that's Down syndrome. Now, there are some people, in terms of sex chromosomes, where there's non-disjunction as well. So, a normal female genotype is XX. Normal male is XY, and that's normal, all on the top. Okay. Now, if a female has an XO, is considered just one chromosome. Then they have what's called Turner syndrome. For the male, if they have an extra X, so it's XXY, they have something called Klinefelter syndrome. And then if you have triple X, that means 3 X's. That's triple X syndrome. And then you can also have an XYY. Okay. Now, most of these different characteristics, for a while it was thought that an individual like this was going to be a bad person, violent, aggressive, all that. It's BS, it's not true. But for a while, way back in 20 years ago or something like that, there are differences in phenotype. I'm not exactly sure what. There was also an idea that if a woman was, let's say, born triple X, problems. They had problems with in the Olympics. Can you let her race against normal 2X women? They're still trying to figure that stuff out. Okay. So just so you know. Now, so there's one major gene on the male, on the Y chromosome, and that's the one that triggers male development. So it's a gene that produces a protein, in this case a hormone, that then results in fetal development for a male, including male brain. So who determines the baby's sex? Is it the mom or the dad? Dad. Okay. So remember, okay, so mom is always going to donate an X, any way you look at it. There's a 50% chance that dad is going to donate an X, in which case they'll have a daughter, or a Y, and then in that case you'll have a son. Now remember, I talked to you about this before. Remember when I mentioned the heterogametic sex? So hetero means different. So which would be the heterogametic sex? Huh? Dad, right. Men, boys, the male. Okay. This is going to be important because if there's going to be any sort of, let's say, mutations that show up, bad mutations, they're most probably going to show up in the males instead of the females. And the reason is, with all the other autosomes, you have 2 complete chromosomes, right? With the same genes but different alleles. So a lot of times, if you have a mutation in one, but you have a normal in the other, then you'll end up with normal. Like, let's say the flower color, and it's dominant. You have red, you have white, RW is still red, RR is red, WW, it takes 2 of those for it to be recessive in a white. Well, with an XY, there aren't 2 identical chromosomes unless it's a female. So that's normal. One female has an X that's got a mutation on it that's lower fitness. A lot of times you'll have a rescue effect. However, with a boy, and they get a problem on the X chromosome, or even the Y, but let's say for X now, there's no rescue effect on the Y. So for the boy, it's like getting YY. In other words, or the flower color WW, because there is no rescue effect. So you're usually going to see that, that phenotype with lower fitness on the male. Now, just so you know, birds are different. Birds have a ZW sex chromosome. And in this case, the males are homogametic and the females are heterogametic. The females are ZW, the males are ZZ. Okay. So these are called sex-linked genes. Males get their Y, of course, from dad, and their X from Can male be carriers of an X-linked trait? Anybody? Yes? No?
Yeah.
Well, if they have it, if they, first of all, if they have the bad X, they can pass it on, that's true, but they also have the disease, right? So we'll say that they're really not carriers. They actually show the X-linked lower fitness phenotype. Only females can be carriers. In other words, they can have one X with a mutation and one not, and they can either 50% donate the one with the mutation or the normal. Okay. So why do some genetic conditions affect males more often than females? Okay. A well-known example. On the X chromosome, there are genes that affect the cones in your eyes. Those are the ones that detect not only light intensity, but color. And if a female has an X that's a mutant X, and she herself will not show color blindness. But if she donates it to a male, that male offspring will. So if a colorblind man marries a woman who is a carrier for the recessive allele, what's the predicted outcome for their children? Okay. So okay. So we'll say okay. We'll use C, large C, and small c. Or we'll go normal and colorblind. There we go. Okay. So the woman is a carrier on her X. So she's normal. Okay. Now remember, these are both X's. So let me, and then up here, the man has an abnormal X. And a normal Y, except that, of course, he's out of it. Okay, so if you have a normal and a colorblind, that'll be this will be that'll be normal male. And if we have 2 X's, this will be, it's never going to normally happen, but and this will be okay. So we'll have a normal male, a male with color blindness, a normal female carrier, and then a female that's colorblind, which never is going to happen. So we can actually cross this out. Okay? Oops. Okay. Same sort of thing. Hemophilia, this is the disease of the royals in Europe. The royals were really inbred. They had all sorts of recessive problems, and they also had sex-linked. And so a lot of the male royal offspring ended up having hemophilia. So hemophilia, that means you cannot clot regularly, and even a bruise will lead to massive bleeding. And so most of these babies didn't live very long. By the way, the royals, by the time of the end of the royals, you know, with the royal houses in Europe and the various kingdoms, they were all interrelated from interbreeding because they didn't want their kings and queens to be married to a commoner, because it was all the divine right of kings to actually, according to God, to rule over people. So by the time that actually ended and there were different forms of government, the royal lines were so inbred that oftentimes a king and a queen who might have been first cousins or something like that might have ended up, let's say, having 10 babies and none of them or only one of them would make it to adulthood. And that one might actually end up being insane. Okay? So it kind of led to a mess. And they were bound to, I guess, topple eventually. So here we are. We've got hemophilia. It's an X-linked trait. If a female married an unaffected male, what fraction of their sons will be affected? Okay. So, we'll say XN normal, and, oh, actually, X colorblind. No, that's not right. Come on. Why did it do that? Okay. No. Okay. So this X up here is, of course, colorblind. So we have an N NC oh. Okay. Oh God, that's what a mess. Okay, so we'll have an X normal female, well, carrier female. Below that, we have an X with the carrier, so we'll have a carrier female here. So this is a normal female up on top, carrier female on the bottom. And then as far as the Y, of course, we'll have a normal male up on top. And then we'll have, on the bottom here, colorblind male. So if you are a colorblind male, which parent gave you your colorblindness? The mother, right? Because it's an X-linked trait. Now there are some genes on the Y chromosome that only males have. And you could have a mutation in one of those and it would only affect males, and there'd be no such thing as a female carrier in that. So XX, XY. Colorblind is on the X chromosome. So it's always going to be, we'll say it's this one is always going to be donated by that one 50% of the time. Now, I also talked to you, we've been talking mainly about single gene changes. And then we'll call them qualitative traits. That means this or that. So if we have purple or white, and purple is dominant, you either have the allele for purple pigment and either 2 copies of that or 1 copy of that and white, and you'll end up with purple. Cystic fibrosis is the same sort of thing. It's a qualitative trait, single gene. It's recessive, so you need 2 copies. That means both of your parents have to be carriers. Attached earlobes are the same sort of thing. Now, quantitative traits. Most of our attributes, our characteristics, our traits, are what are called polygenic. So Okay, that means there are many genes that have a small effect. So like height is affected by many genes, and depending on which alleles that you have, you can end up at various heights, and it follows a normal distribution. So let me give you an example. Okay, so if we look at height, usually when it's a quantitative trait, that means polygenic, it's usually going to follow a normal distribution. Okay, so we've got height. We'll say the average is 5'8", and this shows on the left on this axis over here, y-axis, that shows the number of people in each group. And it's all because of each gene, and there's a lot of different genes that affect height, just adds a small effect. Now, in actuality, this is what we look at when we look at a normal distribution, but in this case, we actually have a distribution like this. It's not actually that much. We'll say okay, and this is called bimodal. Okay, bimodal, it means that there are in fact different, two different averages, and it corresponds to gender. So in normal circumstances, women are generally shorter than males, and so we look at them separately. They each have different means. Women might be 5, 5, and men 5. Okay? So that's polygenic. Most of our genes, many of our genes, and the traits that correspond with them are like that. Okay. So this is called polygenic again. Now, which trait is more likely to show continuous variation, like a normal distribution, a monogenic or a polygenic trait? Like continuous variation, which one? Polygenic, right? Okay. Now, it turns out eye color, I've been kind of treating it as a monogenic, but it's not. Eye color is, there are 3 genes that actually affect eye color. And with these 3 colors, it's basically dark brown, I think I'm not sure what the other one is. anyway, there's 3 of them. So that's why you can actually get a range of eye colors, everything from brown to greenish, light blue eyes, dark blue eyes, like Paul Newman up here was renowned as an actor who had these bright blue eyes. Women kind of swooned and fell over when they saw him. Okay. Now, I've been treating it like these genes, they code for certain things no matter what. And they do. But for the most part, especially polygenic traits, it's always a matter of genes interacting with the environment. So here we have a woman. If she had stayed out of the sun, she'd have smoother skin. If you're working with your hands a lot, you're going to end up with calluses, okay? Versus, now that's all gene expression, genes in that area, and calluses develop. You're not born with calluses on your fingers. They develop if you use your fingers all the time. The same thing happens with your bones. If you end up sitting in a chair all the time versus hauling around rocks, your bones are going to be bigger if you're hauling around rocks. You'll also see some sorts of arthritis usually eventually. So I just want you to understand that genes are always interacting with their environment. Now, there are some plants, and if you were to take they grow at, let's say there's a plant that grows here around sea level, and there's also all the way up to an altitude of 10,000 feet. If you were to take a plant that was always grown at sea level and put it up at 10,000 feet, it probably wouldn't, even though it's actually genetically identical, because the genes are interacting with the environment, those individuals wouldn't do so well. And vice versa, if you took one from 10,000 feet and bred it down here, it wouldn't do so well. Okay? These are the same genes, the same alleles, but they're expressed differently. Okay? Eventually what would happen is the expression pattern would start to change. And that's part of gene regulation that we kind of covered. Okay. So when I say genes by environment, a lot of times you hear people talking about nature versus nurture. In other words, nature, that's your genes. Nurture is your environment. And there were, at one point, looking at human psychology, there were some behaviorists who actually thought it was all nurture, it was all environment. Famous guy named Skinner, he said you could take any human baby and raise it in a certain environment and it would end up with those kinds of skills and that kind of phenotype. Well, he was an idiot. According to him, then, you should have been able to take a dog and in the right environment, teach it how to talk, and dogs don't talk. Now, I know dogs recognize a lot of words, but still, they're limited. Okay. What it is there's always an environmental component in how genes are actually expressed. So like skin color is an example. first of all, for a lot of people, you go out in the sun, your skin darkens, you get a tan. That's because genes are putting out melanin, which is a pigment, a dark pigment, so that protects you from the sun. Okay? Now, there's also, in terms of skin color, genes, different color alleles. So, some people, let's say from Africa, have very dark skin. Also from Australia, if you actually look at the genes controlling skin color, though, you see that they have different alleles. Okay? Okay, so heritability, now, we haven't covered this for a while. If you remember during, we were doing Darwinian snails, there's this whole bit about heritability. So, a lot of these traits are somewhat heritable, like height. It's about 80 or 90% heritable. Okay, obviously you've got a mother and father, and depending on whether you're a boy or a girl, there's a fair amount of heritability in the height of your offspring. So if you have taller parents, you're probably going to have a taller offspring. Sometimes heritability can be really low, like 5%. Okay? In other words, it might be a trait, let's say, that's beneficial in certain environments, but if it's only 5% heritable, it's unlikely that it's going to actually be passed along and evolve. So we've kind of...
Just a quick question.
Yeah.
What about, so would that apply to the big fish or small fish, big bowl theory, where it's like, if we were in an environment with a bigger space, we would be taller than our parents, even though they were shorter than us, or they're a little bit taller?
Yeah. So remember, I talked about Sean and Don, and they were identical twins, and one ended up being kidnapped and sold onto the slave market into Chad, and was brought up as a slave in a food-poor environment. And the other one was brought up with an alcoholic mother, because she went through that whole business with pirates, and a good food environment. And the one in the good food environment, that was Don. He was 6'4", let's say, or 6'2", and Sean, the one that got kidnapped, he ended up being 5'10". Okay, so there's variation in there. It's all because of the environment, the food environment. So if you put both of those from the time they were little babies in the same food environment, they would, and depending on which food environment it was, they would end up at a certain height. Okay, it wouldn't just be, oh, they're always going to be 6'4". No, it's going to be how much food is there, and then what's the, in the best environment, they might actually be 6'4", let's say. In fact, in my lifetime, people nowadays are bigger, taller, than when I was growing up. Now, that's a long time ago. It has nothing to do with the fact that the genes are different in terms of height. It's all gene expression patterns, okay? There's still, some people have tall parents, and some people have shorter parents. So they'll have a certain genetic part of the component that's heritable. And then, depending on what environment they are, in the food environment, you end up with that variation there. Okay, and it gets even more crazy than that. It turns out it's gene. It has to do with gene regulation and marks that are put on to either repress or express genes, and those marks can actually be inherited. So, but you don't have to know that. It's called epigenetics. But anyway, so now we know a lot of this because there have been twin studies that have looked at identical twins, fraternal twins, and then identical twins that have been raised apart. And one of the most famous cases was Jack and Oscar. And there's, let's see, I forget. Anyway, you can look at this. There's actually a little film on it. But there were these identical twins down in Trinidad. You had the father was Jewish, the mother was German, and just as the war was breaking out, mom took, I think it was Oscar, and dad took Jack. Now, one was raised as a Nazi, one was raised as a Jew. This really screwed up with Oscar's head, as you can well imagine. But they also shared a hell of a lot of traits. They used to like to make big sneezes to, like shock people or whatever. They did a whole bunch of different things that is obviously there's a genetic component there. They inherit exactly the same genes. At the same time, of course, one's anti-Semitic and which means he also hates himself. But eventually they managed to put their politics aside. They got brought back together. They discovered each other. It was kind of hard at first, but eventually they kind of developed a relationship. They died recently in 2011, I think, or one of them did. One of them might still be alive. So, but they've done a lot of studies now with twins that have been separated at birth. So, as I've been mentioning, we've been talking about gene by environment interaction. Well, that's how you can actually get at which component is environmental and which component is genetic. Okay? Where you have identical genes, and then you see how they're expressed in different environments. So, with a lot of quantitative traits, especially since these are genes with small effect, it's actually hard to determine what kind of phenotypes. Also, there's differential expression patterns, like which of you looks more like your father? Anybody? You do? Okay. Anybody else look more like their mother? Okay. Anybody look neither like them? Okay. Milkman? Sorry.
Oh my God.
Milkman, that's what they say.
Sorry. Okay. Anybody in here not look like your parents, but like your grandparents? Okay. There you go. Wasn't the milkman after all. So, and that's one of those things. So sometimes, like I look more like my mom. But I know I have some of my dad's genes. So there's probably a mix, but sometimes one parent or another is generally more expressed than the other. Okay?